Welcome to the latest Novogene webinar, Innovative Nucleic Acid Extraction Technology with BioEcho Life Sciences. Today's webinar will be in collaboration with the nucleic acid experts, BioEcho. If you are interested in watching some of our other beginner's guides to sequencing, please visit our website where all our webinars are available to view on demand. We will send you a link to our website in our follow-up email. A bit of housekeeping before we start. The presentation will run for around 40 minutes with 15 minutes for questions and answers at the end. Please submit your questions via the Q&A button at the bottom of your screen. You can upvote a question by clicking the thumbs up symbol next to it. We will try and prioritize the most popular questions. If we don't have time to answer your question, we will come back to you via email after the webinar. Please don't use the raise hand functionality. Just send any questions via the Q&A or message us directly if you have a technical issue. We will share the slide deck and a recording of the webinar via email. You can expect this on Friday. If you are studying and need a certificate of attendance, there will be a link in the follow-up email that will display the certificate you can print from your browser. Finally, there will be a short survey that pops up when the webinar finishes. Please take a couple of minutes to complete this as it helps us to plan future webinar topics. I would now like to introduce your host for the webinar, our Product Marketing Manager, Yue Zhou. Yue, over to you.
Okay, thank you, Miranda. Okay. Hello, everybody, and thank you for joining our webinar today. And today's topic is, as you can see, about innovative nucleic acid extraction technology with BioEcho Life Sciences. So my name is Yue. I am Novogene's Product Marketing Manager and the host for today's webinar. And shortly, I will pass over to Nika, who will talk you through the principles behind each method available for nucleic acid extraction and the various tools used. Also provide tips for optimizing the purification process for downstream applications. Quality control and safety considerations will also be discussed. So I appreciate many of you are probably customers of Novogene already, but before I hand over to Nika, I'm going to speak a little about us. So just in case there's anyone here joining us for the first time. Okay, so Novogene was founded in 2011 and has been growing rapidly since then. Our company mission is to advance genomics and improve life. And as you can see here, our services cover a huge range of applications with real-life implications, from complex disease research and oncology to food and water safety. Our European Sequencing Center is based in the Cambridge Science Park in the UK. Here we have our ISO 17025 certified and also state-of-the-art lab equipped with Illumina Nova 6 sequencing machines. We have always provided fast turnaround time and also high quality data, but that has been improved even further with the installation of Falcon 2, our new intelligent sequencing platform, which has increased our monthly capacity by 200% compared to the manual steps. We also have our supercomputing cluster, so we can offer bioinformatics services and store your data in the UK. However, at heart, we are a global company. As you can see, we can offer services across Asia and US and of course Europe. Falcon 2 has now been deployed across a number of our labs, standardizing our services globally, with almost 2,000 employees and serving over 5,000 customers in world-class universities, biotech companies, and hospitals. We can provide sequencing for the smallest or the largest projects. So we provide a full range of NGS services such as whole genome sequencing, whole exome sequencing, and various types of RNA sequencing, metagenomic services, and epigenomic services. This can be applied to a wide range of research topics, But today we'll be talking about nucleic acid extraction, which is starting point of all of this. And as you know, many other companies, they offer their services too. So why Novogene? There are a lot of good reasons why you should trust us with your project. As I mentioned, we have had a really exciting development recently in the installation of Novogene Falcon 2, our new intelligent NGS delivery platform. And this has now been deployed in our Chinese, USA, and Cambridge labs, standardizing our global service. And this new technology was developed in-house by Novogene and has already massively increased the capacity of the Cambridge Sequencing Center. It will also increase the quality of your sequencing data as it is entirely automated and will remove any possibilities of human error. With this new addition, we are now able to offer our fastest ever turnaround time for RNA-seq and whole genome sequencing, just 10 working days from sample arrival to data release. So Falcon 2 really highlights the innovation we strive for here at Novogene in an effort to keep pushing the frontiers of genomic research. So the benefits of our in-house innovation are clear in our record in the scientific community, which we're very proud of. We have been cited in over 790 research papers with a total impact factor of more than 6,000. We're also driving the NGS industry forward with over 37 NGS-related patents. And our technology is helping to make the NGS process more efficient and effective, and all the while improving the quality of the data we deliver. So the NGS, the Novogene community keeps on growing, and our expert sales team speaks over 10 languages and is located across Europe, ready to assist you in your next project. So if you're not ready to send us any samples yet, you can still benefit from our educational outreach to learn how NGS can work for you and also for your project. I would like to just quickly introduce you to our European sales team, and they are based across the continent and ready to help you with your next sequencing project. And if you would like to speak to your local sales representative, you can email us or simply just leave a message during this webinar, and you will hear from us and from them. So finally, as you know, our presenter today, Nika, is from BioEcho. So just for those who are not familiar with the name yet, let me just introduce you a bit about it before I hand it over to our presenter. So as the nucleic acid experts, BioEcho was founded in 2016 and headquartered in Cologne, Germany, and they are experts in molecular biology. They also have their own products, Acolution, which are the fastest nucleic acid extraction technology, and Nika will tell the story behind this later in her presentation, okay? And also, from what I know, they're quite passionate about the sustainability. So with that, I would like to thank you for your attention and will hand over to Nika. And Nika is the application specialist at BioEcho and is an expert in all things relating to nucleic acid extraction. So with her expertise, she will guide you through the history, methods, principles, and also applications of nucleic acid extraction. Okay, over to you, Nika.
Thank you so much. Give me a second to share my screen, and then we are all set. Here we go. Yeah, thank you so much. Also from my side, in the name of BioEcho Life Sciences, I want to welcome you to this joint webinar. We are happily working together with Novogene regarding nucleic acid extraction services, sharing thereby our expertise to fulfill your requirements from nucleic acid purification up to sequencing results. And we are very proud of this collaboration. So I feel very honored to have the chance to speak here in front of you. As you kindly introduced, I will take you today with me through the mechanisms and principles behind nucleic acid extraction and purification technology. My aim is to learn together the methods available and get more insights into these workflows. So we will unlock the secrets of nucleic acid purification, or let's say, learn how to become a nucleic acid expert. And I really want to say here that being an expert does not mean that you know everything on the planet, because I mean, I also don't. I always realize that there's so much more to explore, especially in the life science world. However, I try to summarize in this presentation all my knowledge about nucleic acid extraction and purification for you. So whenever you see these two little mascots of BioEcho, I try to give you some tips and tricks how to extract nucleic acids successfully. Before I start, I want to show you this picture here. This is a proud Atlantic salmon swimming free in the sea. But why am I showing this fish when I want to talk about nucleic acids? And who is this guy here? And what the heck does I want to tell you with this picture? This is Tübingen, by the way, a very nice town in the south of Germany. If we combine the salmon, the picture of the guy, and Tübingen together, we end up with the first isolated DNA molecule, which was called nuclein. So I'm firmly convinced if we are talking about DNA and if you're in genetics, you probably do not think twice about when DNA was discovered. So in 1953, DNA structure was solved via X-ray by Watson, Crick, and Franklin. This a lot of people know via textbook or via that studying. But earlier, in 150 years ago, back in 1869, DNA was isolated by the Swiss biochemist Friedrich Miescher in Tübingen by salmon semen or via salmon semen. This discovery Miescher made laid the foundation for our daily business dealing with nucleic acids and for the central dogma of molecular biology. This dogma states that if we understand a gene encoded by DNA, we could also understand the function of the gene, means a biological function. Miescher was not alone. So I listed you some milestones, no worries, we do not go to every milestone here, but I want to show you how the discovery of nucleic acids started and leads us to maybe an important invention from our daily life. So probably there are a lot of other important events which I missed here for reasons of simplification, but this does not mean that those discoveries are not less important. But as I mentioned, so the history started in 1866 by the discovery of Mendel and his crossing experience with peas. And we named him in biology the father of genetics, followed by the identification of nuclein by Miescher, the DNA structure published by Watson, Crick, and Franklin, and also by the statement of the Chargaff rules, which state how the nucleic bases are distributed in the DNA molecule. In 1955, it started with the discovery of nucleic acid purification methods, started with size exclusion and anion exchange chromatography, quickly followed by all the other methods which are still established in our labs, kind of maybe by homebrew techniques, but also included in commercial available kits. And this shows that a lot of things were going on when once the DNA molecule was discovered. And I think this is quite important to understand that the techniques we are working with today are quite old, but they're still working pretty well, as you all know. I do not want to bother you so much with history because we wanted to talk about a totally different topic. For me, the question is more about why do we need to talk about nucleic acids and its purification? So as I said, the central dogma states that information is stored somewhere in the gene and encodes for function. As a consequence, if we understand the chemistry behind nucleic acid and its function, if we understand those tools to investigate nucleic acids, we could solve biological functions or even important tools for medicine, like this famous topic we were all suffering from during the last years. But I mean, being a scientist does not always mean that we all have to rescue the world by inventing vaccines. I'm very confident that most of you are working with nucleic acid isolation because of dealing with applications like PCR, cloning, sequencing, or even third generation of sequencing. So nucleic acid isolation is our daily business in life science and life science industries. And if we investigate nucleic acid, this helps us to solve biological scientific questions. So means that getting the pure nucleic acid and studying its sequence, for instance, is the fundamental process to get to our answer about every biological question. Why could this be tricky? So nucleic acid purification challenges us. I'm firmly convinced that we are all working with totally different input types. So probably you are working with this or something like this, or maybe those sample types. So no matter what it is, all of us want to isolate in the end some kind of nucleic acid to solve a biological question. What is the bottleneck here? Doing your experiments, finding the answer, and finish your project with success. It's the quality and the success of your nucleic acid prep. So imagine you do not understand the process behind your purification, and then you got kind of biased results like this. So imagine you spend months working on a construct, do your cloning experience, and get a positive clone. Do your DNA isolation, send it for Sanger sequencing, and you're getting those files back. Disappointing. Or you got rare human tissue, spend some hours on RNA isolation, and got the bioanalyzer greets you with this nice electropherogram saying, sorry, DNA contamination here and here. Those scenarios could be frustrating and could lead you into depression and loss of self-confidence. We all know these situations in the lab. So the solution could be to start at the bottleneck, start at the nucleic acid purification to avoid garbage in and garbage out results, which leads you to depression. First of all, if I consider to do a nucleic acid purification to solve my biological questions, I have to understand that I have to deal with requirements and challenges. Requirements could be a simple, rapid workflow, prefer dealing with harmless reagents because I don't want to get in touch with toxic chemicals usually. We want that our downstream applications succeed. We want reliable, reproducible data. What could challenge us? It's not only that we have to deal with several different tissue types, could be easy to lyse, but also hard to lyse. Could be that our nucleic acid extraction, nucleic acid type we want to investigate is low concentrated. We also have to deal with challenges of our project. We want to create value. We want to stay in the timeline. Maybe we have to stay in the budget, or we have some stakeholders like our professor waiting that our PhD project is finished soon. So I want to conclude and to underline that if we understand all these challenges and requirements, if we understand what we want to address, we could start with our purification much better and leads us easier to success. Usually I'm saying that nucleic acid purification is a mission of 2 quests. So I divide the whole process in 2 big quests, sample extraction and nucleic acid extraction. What does this mean? Sample extraction means that we first have to release our nucleic acid from our sample. Could be cells, we have to break down a nucleus. Could be also that we have tissue and we have first to disrupt the tissue to even get to the access to the DNA or RNA. The challenge is here that we have to find the best method to homogenize our sample. The second quest is then the nucleic acid extraction by itself. Means that we have now our disrupted tissue and we really want to get the DNA out and to get rid of all the other stuff. By dividing this whole workflow in two quests, helps us to solve the sample question first and then go to down to the molecular biology, to the real chemistry to isolate our nucleic acid of interest. And if we solve both quests, we rescue our NA, hopefully successfully in a high pure quality and with a high yield. Let me first start with the sample extraction. So our challenge, as I said, is homogenize tissue, cells, break down cell walls, maybe, without losing our nucleic acid. Extraction could be done via chemical disruption, via enzymatic disruption, but also via mechanical, physical disruption. Probably you somehow combine all these three processes. The goal in the end is, as I said, we want to get rid of tissue. We want to break down cell membranes. We want to get access to nucleic acids without fragment our nucleic acid by itself. So chemicals could be helpful to extract DNA or RNA. They could be used alone for easy to lyse materials, such as tissue culture cells, or in combination, as I said, with other methods. Thereby, cellular disruption could be accomplished by agents such as charotropes, strong bases, or even detergents. Enzymes could be proteinases or proteases, I think most of you are familiar with, but also lysozyme maybe for gram-positive bacteria. You also have to be aware of nucleases coming from the tissue. So it could be beneficial if you want to isolate DNA to add an RNase to digest the exogenous RNA to just have a pure sample with gDNA resulted. So physical methods for nucleic acid purification involve always mechanical disruption, which is not super specific, but very efficient. So this method could be freeze-thaw cycles, which work perfectly for cells, but also bead beating or sonification could be a preferable method. So if you want to isolate maybe mitochondria, I could recommend you sonification as a process. Sample disruption is an art. So if we consider, for instance, bead beating, it's not that easy to find the best bead for your sample. So I try to give you here some advices or rule of thumbs. So if you have larger samples, bead beating is quite a nice method to disrupt your tissue. Glass beads could be used for prokaryotes or yeast, and stainless beads for animals or plant tissue. And if you ever combine all these methods together, chemicals, enzymes, and also the mechanical physical method, you always have to consider the compatibility of your lysis buffer, your enzymes, that they all could work together. I have to admit that sample extraction is a real art, as I said before, and I wanted to give you some additional advices. So in particular, you could divide samples into two groups, easy to lyse and hard to lyse tissue. Easy to lyse tissue could be culture cells, ear punches, blood, for instance. Exosomes, viruses are quite easy to lyse. So usually they are just cells with membranes without cell walls, and they come with a liquid input. As I said, you can disrupt them easily by freeze-thaw cycles or via detergent or enzymes, or sensitive grinding could be helpful here. Hard to lyse tissue could be sometimes a challenge. So this could be plant-derived tissue, means cells with rigid polysaccharide-rich cell walls, or gram-positive bacteria, insect cells, not that easy, or yeast, or complex tissue. There it could be very helpful to do your bead beating on dry ice or with liquid nitrogen, because you always have to be aware if you do a mechanical disruption, you put pressure on the tissue, which creates heat. And heat usually is not that what, for example, RNA likes. So sometimes bead beating under cool condition could be very helpful here. But also here you have to keep in mind which buffer are you using, because some buffers get super viscous when you cool them down. If we solve then our sample extraction method, we have to move on with quest number 2. So the nucleic acid extraction itself. This means that we want to recover our nucleic acid from our sample. The challenge is here that if we break down a cell, there's not only nucleic acid inside, there are proteins, lipids, cell debris, everything which could kind of affect our downstream application. So we want to get rid of, let's say, all these contaminants and without losing our nucleic acid. We can divide this nucleic acid isolation in 2 different approaches. Means we can divide it into liquid-based extraction and solid-based extraction. What does this mean? So liquid-based extraction are always methods where we interact with the liquid, and usually it follows the principle of solubility. It could be an organic extraction like phenol chloroform, or an inorganic extraction when we're dealing with ethanol precipitation, for instance. Solid-based extraction means that we have a solid phase where our lysate, let's say, interacts. This could be gel filtration, ion exchange chromatography, and affinity chromatography. Thermal extraction could be also here helpful. Maybe if you want just to check for bacteria and positive clones, then you can do your direct PCR, means do a thermal extraction, heat up your bacteria colony and put it directly into your PCR. Drawbacks are here obviously low quality and purity, because we do not involve any extraction method. So let me start with the liquid-based extraction. So the easiest method is precipitation. It's based on the principle that we neutralize our nucleotide, we decrease solubility, and we pellet then the nucleic acid and resuspend it. You can see it here. So we succeeded with our sample extraction. Then we neutralize our nucleotide by adding salt and alcohol. We precipitate our DNA, and then we resuspend it in tea or water, and we got our DNA recovered. Benefits could be here that it's fast and very cheap and does not require any commercial kit. It's very flexible and leads you to high yield. However, it's not really specific, because it's just works with solubility properties. So it's temperature dependent, and usually it does not get rid of contaminants, means lipids and proteins. The precipitation method was kind of invented a bit more, and oh, I missed here something, which is very important. One tip of mine: if you want to elute smaller fragments, with this method, you could also add magnesium chloride to increase your yield here. The precipitation method was integrated in the most, let's say, famous method, the phase separation. As an example, the phenol chloroform phase separation, which I think you are all firmly convinced with. So this principle based on phase separation. So what we have our sample, and then we do a phase separation by adding a phenol chloroform mixture. And by centrifugation, we got these 3 phases: organic phase, the aqueous phase, where our nucleic acid is present, and an interphase with residual proteins. We do then the precipitation I presented you before, could be in a tube, could be also on a column, means a column-based extraction then, which leads us then to our eluted nucleic acid. This is a gold standard. It usually leads you to high yield, and it's commonly used if you have large input. So if you have large input, you could consider this method. It's flexible to isolate RNA and DNA, and it denatures proteins rapidly. So we get rid of these contaminants quite easily. But you're dealing with toxic reagents. So means that we kind of have a risk for the experimentator, but we also have to manage the waste we produce here. So if we have a lot of reactions, so we also have a lot of toxic waste. It could be laborious because this phase separation could be sometimes not that easy. It's not that feasible for automation, and sometimes it also leads to low purity because if the phase separation was not super successful, we have a phenol carryover, which then kind of interacts with our downstream applications. As I said, it's quite flexible to isolate RNA and DNA. And I have a little trick in the lab. So if you adjust the pH a little bit, it's easier to get, or let's say enrich, for instance, DNA or RNA in the aqueous phase. What is also nice, if you have a real protein-rich sample, then I could recommend you to, if you are in this phase before phase separation, do a nice and very rigid vortex step before phase separation, because then the proteins really remain in this interface. Finishing this liquid-based extraction, I want to move over to the solid-based extraction. The most easiest principle is gel filtration. How it works. So you have a stationary phase with a selective pore size, and then you have a mobile phase. Could be your sample. We're starting with our sample extraction, and then we apply our lysate on our gel filtration, and by gravity, for instance, molecules get separated, and usually bigger fragments get eluted, whereas other contaminants, smaller molecules, remain in the matrix. This is a nice method to get a low-cost isolation. Could be fast, but could be also take long time if we consider gravity or centrifugation, but it's low cost and it has less hands-on steps. Drawbacks could be that if we don't understand our lysis conditions and also this stationary phase, that we have an interaction between the liquids, but also our nucleic acid and the stationary phase. It's limited to volume, depending on the column size we are using. But it's very efficient to elute large molecules, because if we have less centrifugation steps, this could lead to less shear forces. So if you're interested in bigger fragments, then this could be a nice option for you. Another method, most famous, is our spin column based method. So I think a lot of you dealing with spin columns. So spin columns follow all the same principle: bind, wash, elute. Could be, depending on the sample, also several washing steps. So we are starting again with our extracted sample. We applied it to our column, our spin column, which are usually commercially available. And then we centrifuge our lysate through the matrix, and the flow through you can discard. And then we have the binding step. So we hope that our nucleic acid, in that case DNA, is bound to the matrix. Then we wash with several buffers. Usually they are commercially available. And we get rid of contaminants such as proteins. And then we elute our pure DNA. The principles are usually based on affinity chromatography, but also ion exchange. So the famous example are the silica membranes. Usually this leads you to high pure nucleic acid. It's easy to perform, reproducible. And depending on what you're doing, if you're doing it by yourself, you can sometimes reuse the resin, which is, I think in the modern kits, not the case anymore. You can centrifuge it, but you also can use vacuum. It depends highly on input, because if you have this binding reaction, this could mean if we put too much input on site, we kind of the binding affinity of the column is totally saturated, and we lose a lot of our nucleic acid. High salts and carotrophs could be carried over, so means that our eluate could be full with high salt and carotrophs. And depending on what you're using, usually this method based on carotrophs, such as ethanol, you have to also implement waste management, because usually carotrophs could not be wasted in the sink. This method is quite nice if you want to bind smaller RNA fragments, for instance, or elute them. So if you once have the chance to work with this method and you think about, oh, I want to enrich smaller RNA fragments, then I could recommend you to increase the alcohol content in the binding buffer. And this helps you usually to bind smaller fragments, means microRNA, for instance, which you can then elute by following the principle. Another solid-based extraction method is the magnetic bead-based method. It also based on the bind wash elute workflow. So, and usually what we have is that we do this magnetic separation by using a magnetic field external, and we have these paramagnetic particles which are added to our lysate, and then we bind with a magnet, like you can see here, we bind with a magnet our DNA. We can then wash the flow through away, for instance, or the supernatant, let's say, or the contaminants. And by repeating this step means put the magnet on, kind of tackle or bind the DNA, discard the supernatant, and then wash and bind and wash and bind and so on. Then we usually get easily to our not eluted, but purified DNA or RNA. So this is a quite nice method to kind of elute your nucleic acid of interest without centrifugation. And by that means that it's also easy to automate. You usually require less equipment. You just need this magnetic rack and some pipettes. And it's adjustable to different input because you can play with the binding material, which is not the case in columns. Because if we add more beads, then we can also bind more input. You do not get risk of clogging, because for a column could be if we transfer also cell debris that our column gets clogged and we do not elute our nucleic acid, which is here not the case. Drawbacks could be that beads usually cannot deal that great with viscous samples. And if we did not adjust our binding conditions perfectly, could be that we also have bead carryover, and this interferes then with PCR, for instance. If you're not familiar with bead beating, with magnetic bead extraction, you sometimes kind of get confused by this binding and get rid of the magnet and so on. So that's why I said that it requires experience and could be sometimes, if you do it hands-on, laborious. So as I said, if you want to increase yield, you can also increase the input of the beads. However, it's kind of not that easy to find the spot with means that you have to find the right ratio of beads and your sample. But if you want to increase the beads, the yield, I could recommend you to perform a sensitive inversion of your sample, means turn it a little bit upside down during binding and give them the beads a chance to bind your sample. So now you heard a lot about different nucleic acid purification methods, and maybe you realize that these methods are quite old, invented in kind of starting 1950 up to the 80s. But maybe you realized already in the lab that the downstream applications evolved quite fast. So since 1953, when DNA was discovered, a lot of things were going on. And today we talked about third generation sequencing pursued by Novogene, for instance. But what happened with our nucleic acid prep technologies? Simply nothing. So we at BioEcho had the idea to, let's say, fill this gap with our innovative technology, our Echolution technology. And by inventing this technology, we made nucleic acid purification faster, simple, more reliable, and also sustainable. The workflow is quite simple. So we have our sample diversity, let's say, and start with the lysis. After this lysis, we can directly transfer this lysate to our purification matrix. And only by one single step purification, we elute our inhibitor-free, ready-to-use nucleic acid of interest, whereas all the other stuff remains here, or maybe also here on the matrix. If we combine this workflow to the established methods I introduced you before, you can here clearly see that by this single step purification, we reduced hands-on step. This is an example of our RNA isolation. So if we compare our ecolution technology to bind, wash, elute method, we were able to reduce this bind, wash, elute workflow only to an elution workflow, having this lysate, single-step purification, elution workflow. And this makes our nucleic acid isolation faster. It comes with less hands-on steps. And considering RNA, less hands-on steps means also less risk of contamination. And it also reduced chemical waste, because if we do not need all these washing buffers, then we do not have, in the end, a lot of liquid waste. To convince you about this, I brought you an example here today. And here we compare a silica-based DNA extraction kit with a DNA extraction kit with the ecolution technology. You can clearly see that we definitely reduce the plastic consumption. And which I said, we also were able to reduce the liquid waste up to 5 times. Don't let yourself get kind of confused by this coloring. It's obvious that the most of silica kits do not come with blue color, and also our kit does not come with orange, but otherwise you cannot see the difference here. So it means that using also this method does not mean that you are faster and maybe get rid of all these disadvantages of other purification methods, but this is also a choice of sustainability and also many other positive factors. Getting back to where I started, why nucleic acid purification and the understanding of the mechanisms behind is very important. I want to remind you about this. We want to avoid garbage in and garbage out results, which means that if we got these results, something indicates us, oh, went something wrong. Could be impurities, could be low yield, could be some other cases of bad quality. So impacts of impurities, I have to admit, is usually yourself. Impacts of impurities could be divided in handling, sample, and nucleic acid purification itself. So from handling, sources could be you, as I said before, means maybe you do not wear gloves and you have your RNases on your skin and then contaminate your RNA sample with your own RNases, and then all your RNA got disappeared or fragmented. It could be storage. Probably if you're dealing with tissue, you got sometimes tissue in such a bad quality with kind of freeze burn or something like that, or transport because somebody forgot to put your sample maybe on dry ice. From the sample, it could be cell debris, protein carryover, nucleases, which degrade your nucleic acid of interest, DNAs, RNAs, lipids, and secondary metabolites, usually a source in plants. From nucleic acid purification, contaminants could be salts, carotrophs, phenols. Also the pH in the elution buffer could be sometimes an issue and does not match your downstream applications. So I wrote you here some impacts down, which I mentioned before, and some consequences. So if we have a contamination for sequencing, this is a pity because usually it spends a lot doing the DNA isolation or the RNA isolation, send it for sequencing, and then somebody does this analysis, and you know that this analysis is not that easy. And then you got this results back, reading bias, incomplete, inaccurate, ambiguous base calls. Or for gene expression analysis, you do your RT-PCR, and then you got not reliable results. Cloning, low efficiency, no clones on the plate. For diagnostic, also a drama. So if you got wrong false positive or false negative patients, such for COVID-19 testing, and then you have to stay home and you're not sick and not effective. For oligosynthesis, also not the best. All these stuff will end you up in, could be wrong diagnosis, loss of money because of the project, loss of fun, loss of confidence, and leads you to depression, as I said before. So how do we measure nucleic acid quality? This could be by photometric measurements of nucleic acid, and this based on absorptivity properties of nucleic acids. So means that we measure the absorption spectrum of our eluate, and we got some parameters and benchmarks, which I listed you up here. Advantages could be that it's simple. You can directly measure your sample, and you get also an information about the purity by investigating these absorbance ratios. You got an information on contaminants, depending if you have a contaminant which absorbs, for instance, protein as 280 nanometers. Drawbacks is that it's not selective. We cannot distinguish between DNA and RNA. It has a limited sensitivity, and it's easily affected by pH, for instance. And it's sequence dependence, because all this absorbance depends on the sequence of the nucleic acid. Another method could be fluorescence. By that, we usually have a fluorescent dye or fluorogenic dye that binds selectively to DNA or RNA. And it's more accurate than the photometric measurement, and it can be used to distinguish between different nucleic acid species. So what it does is that we bind our nucleic acid, and then we got, by the signal of the fluorescence, we can calculate the concentration of the nucleic acid. It's specific, it could be sensitive and very accurate, despite if we have a contamination. But it could be time consuming, because you have to prepare the dye and the aluate together, and maybe dilute your aluate. And it does not give you any purity information. Fragment and integrity analysis is kind of the well-established and acceptable quality control method also for sequencing. So probably if you're dealing with RNA, you know these nice graphs here. This is electropherogram and shows this gel build. So this method based on electrophoresis, digital. And it usually gives us some quality parameters like the integrity number for DNA or for RNA. Also, the electropherogram could be quite helpful. So we usually say that the 20S, 18S ratio should be 2:1. Advantages are here that you have a ready-to-use device, the screen tape device, for instance, which you have to buy, which is then a disadvantage. But it's universally accepted as QC for NGS, for instance, as I said before. So if you do this method, or your NGS facility does this for you, you can be very confident about the quality of your aluates or samples. As I said, you need the device, and sometimes the maintenance is quite expensive, and you need to buy your kits you want to use. You heard a lot about nucleic acid purification and its pitfalls. I think the most important thing is to keep your nucleic acid single. And this is what I want to illustrate you here. So means if I want to isolate RNA and do RNA-seq, get rid of your DNA. No matter what you want to do, get rid of your DNA. Maybe by DNase digest, whatever you want to do, but keep your nucleic acid single. I listed some additional points up, which hopefully help you. I named this bag of tricks. And I want to, let's say, summarize this quite briefly. So DNA isolation and RNA isolation, you can read it later on. It's very important that you know your sample. You protect your nucleic acid. Could be by enzymes, could be by chemicals. And wear your personal gloves and avoid aerosols. For RNA, keep things cold, grind tissue maybe on dry ice, and store also your aluates in case of RNA cold by -20 or for longer, -80. I want to conclude or to summarize all these things I presented you in 2 additional slides. In my eyes, the most important thing is know what you want and guarantees your success. Plan your experiment. Take your time before doing any experiments, because then you won't get desperate in the end. You always have to deal with purification method, your application, your nucleic acid of interest, and your sample. So it's kind of an interplay with all of these points. They're all somehow influencing each other. So keep in mind which nucleic acid you want to isolate. What is your downstream application? How much do you need from that? What is your sample? And with which challenges does this sample comes, and which purification method you want to use, you have available, for instance. And then again, can I do my application, my downstream application with this purification method? You always have to consider this. As I said, it's not that easy to find the right purification method, but if we get all this together, I think we can, let's say, downscale all the things I try to explain you in 4 basic questions, 4 key questions, let's say. Means, what type of sample do I have and how much do I have? Which type of nucleic acid do I want? Is it cfDNA or is it gDNA? For what do I want to do with it? What is my aim, my goal? Downstream applications, or do I just want to do cloning experiments? Do I want to do sequencing? How do I achieve this? Means kind of going all these circuits around until you find your perfect nucleic acid isolation method. By that, I want to finish my presentation. And I want to thank you so much that you joined me today. And I'm very honored to be here. Yeah, I want to show you again our company. So as I said, for us, nucleic acid isolation is kind of a passion, but also sustainability. But as I said, being an expert is a constantly becoming process. So we are only at the beginning of our journey. And I'm happy that we have the support of Novogene working with us together.
Okay.
So, yeah.
Yeah, thank you, Nika, for the nice presentation. And now we are here at the Q&A session. We do have many interesting questions. So if you don't mind, I will just start from the first one from Alexi. So Alexi is asking, could you give some more details about how ecolution works? For instance, what is physical chemical basis that allows ecolution to avoid washing steps while still getting pure nucleic acids?
Okay, I hope I got your question right. So I think we have to divide it in 2 processes. So sample extraction means this chemical disruption could help to improve the results of your isolation method of choice. But our technology is somehow independent of this disruption method. So means that we can also, we cannot apply a kidney on the purification matrix, obviously, but means if you kind of disrupt your sample and still have some nucleases inside, our kit helps you to get rid of that.
Okay, thank you. And also, we have one question from Rensk asking, is it possible to automate the BioEcho extraction method?
Yes, it is. We are working together with some liquid handling systems such as Hamilton, and we already implemented some automation processes of some of our kits. If you are interested in that, I think there are also our contact data included in this presentation. Feel free to contact me or our, let's say, contact email address, and we will help you.
Yeah, we still have one more question from Sandra regarding the ecolution product. So Sandra is asking, do you have any input material limitations of the ecolution kits which have an effect on the purity of the eluted DNA? For example, if starch-rich materials still suitable to be isolated with this kit, or is there a limitation in the amount of starch input is there a chance that starch will be eluted with the DNA?
Okay. Okay, got it. Maybe we can divide it into two different questions. So first of all, I want to target this starch issue. Yeah. So carryover of starch from potato, for instance, usually doesn't make you happy. So our kit works very well. We have a plant DNA extraction kit, which works pretty good with those samples, and we are very well experienced with that. So usually we do not have an issue with that. But yes, depends on the input, how much sample we have. We have some limitations here, but usually it's the range of all the other kits. If you are interested more in that, as I said, just contact me and we can discuss that. But usually we are quite well experienced with potato samples, for instance, and it works very good.
Okay, I think we also have some more, let's say, specific questions regarding some materials. So this one is from Agnes, and Agnes is asking, what would be your recommendation of extracting DNA from plants known for the isolate DNA containing contaminants? For example, secondary compounds like polysaccharides, polyphenols.
Okay, so could be two things. If your DNA is already isolated, I would kind of recommend you, let's say, another purification step, means a purification kit. Get rid of these contaminants. If you want to start with your material and know that I have a lot of polysaccharides inside, I would recommend you to check the input ability of the kit, how much input this kit kind of could be used with. Or I would look for a kit with a precipitation step or a phase separation step, because this usually helps you to get rid of those polysaccharides without losing your nucleic acid.
Okay.
I hope this helps.
Perfect. Okay, one more question from Sandra. So what is your experience, or do you have knowledge of differences of elution of sizes of DNA between the different DNA isolation methods? Is the difference of elution of big sizes of DNA, degraded DNA, or even single nucleotides between the DNA isolation methods?
Okay, yeah. Maybe we can start with that. So some of these methods prefer to elute larger fragments. So if you have less centrifugation step, this usually leads you to larger fragments. But it also depends on the sample disruption method, because if we disrupt our sample quite hard, then it could be that our nucleic acid get also fragmented. So usually the affinity means the solid base extraction method do not distinguish between a single strand and double strand. But if you consider that this affinity chromatography could based on charge, let's say, and we have a double-stranded molecule, then binding affinity could be much higher than for a single-stranded molecule. And this you have always to keep in mind. If you want to have larger fragments, I would prefer you to use, let's say, maybe a bead-based kit, or let's say a kit which avoids or reduce centrifugation steps.
Okay, thank you. Also, we have one question from Jane, I think is more specific regarding your extraction service. So Jane is asking, should I use DNA or RNA protect reagent for the tissue before I send it to you?
Yeah, you could do that. I think so that could help, depending on how you want to ship your tissue. If you want to ship it on dry ice, I mean, we are quite flexible. So whatever you have in mind, just text us and we'll discuss this. But usually we can also work with stabilization buffers. So no worries here.
Perfect. Also, one question from Carolina. Carolina is asking, what would be your recommendation to purify RNA, column-based method or enzyme-based method?
Okay, maybe we have to differ here again. So enzyme-based means that we add enzymes which are doing something. means they digest, for instance, endogenous RNAs. But by that, we still have, let's say we disrupt a cell, we have our RNA and our RNAs, and we do this enzyme digestion, then we still have all the cell debris around. So I would recommend you to combine the enzyme-based disruption method with the isolation method. Could be spin column-based, magnetic bead-based, or with our technology.
Okay. Also, we have one question from Peter. Peter is asking, I got a perfect A260 to 80 for my extracted RNA, but the ring value for the RNA sample is only 4.5. Could the RNA be used in the NGS sequencing, and how can I improve it?
Well, I think for the sequencing, maybe you can add here something. But I think so. First of all, I wanted to ensure if the sample quality before starting with the purification was right, because for cells, a RIN number of 4 is quite low. I expect something of 9 and above, or 8 and above. For FFPE samples, it's a very good RIN number. So this is maybe helpful for you not get super depressed. I think I would contact your NGS facility and ask for them. And as you said that the 28 ATS ratio is fine, if I understood it right, then I would say if it's a tissue sample, I would give it a chance. What do you think, you?
Yeah, I do agree that it is important to see what the starting material is, if it is FFPE. And if it's just normal tissue, I think we definitely need to optimize the extraction process to see if any possible degradation happens during the extraction. But if it is FFPE, I think it's okay to be sequenced.
Yeah, maybe to add, because there was also how can I improve it, and I don't want to miss it. Probably it's the homogenization method. Maybe the tissue got too warm, or you kind of spent too much time that it took too long. And I think here we can also try to improve that, but also storage, that if you're done with your method, directly store your eluate, or depending on the kit, maybe do the whole isolation process in a cooling area. Depends also if the buffers like low temperatures.
Okay. Thank you. We have one last question from Paula. And I guess that one is for me, for Novogene, because Paula is asking, should we send blood samples in packaging tubes? So as you know that for us, for Novogene, we do have extraction services, but sometimes we do have some specifications for the tubes types. So for RNA extraction from blood, now our specification is for the packaging tubes. But if you do want to have a more flexible choice of tube types or any other information, we do have BioEcho here to help us, to help you as well. And I guess that's it for all the questions for the Q&A session. Could you please go to the next page? Sure. Yeah, thank you. So I would like to thank you all for your time today. We hope you enjoyed the webinar and found it useful. And thanks for all of your questions. And as mentioned, if we haven't managed to answer your question, we will get back to you via email next week and with the answers. And in the meantime, we will also email you a link to the webinar recording and also the slides, along with a short survey that we would appreciate you taking a bit of time to complete. And the survey will help ensure that we make sure our future webinars will be even more relevant to you and your research goals. And there will be a link in the follow-up email for you to view the certificate of attendance if you need one. And in the meantime, please stay in touch with us if you have any upcoming NGS projects or extraction projects. Now it will be a great time to contact us as well. And you can follow us on social media, or you can get in touch with our website. And also, as you can see, the contact info on this slide. And our webinar next month will be about a beginner's guide to NGS applications in DNA research. and it will be presented by our product marketing manager, Rait. And please do not forget to register if needed. And we will be looking forward to seeing you all on the next webinar. And thank you all for coming, and see you next time. Bye-bye.
Bye.
Bye.